Okay, then, now everything is set up. Good morning to everyone. Welcome to our today's webinar, which is organized by NextGen Biopest, Savva, and the Microbiopest projects. Thank you once again for joining. More people have joined, so we can start now. Just to inform you that this webinar is being recorded, and it will be shared through our project's YouTube channel. So you will be able to find it after the end. And we can start now. Dr. Suad Ruiz will start with some opening remarks. Then I will briefly explain the objectives of today's webinar, and then we will start our session with a brief quiz. You will be provided the QR code, then you will be able to join the quiz, and I will start the questions. So, Dr. Suad Ruiz, the floor is yours.
Thank you, Angeliki. Good morning for everybody. My name is Suad Ruiz, head of the Laboratory of Biopesticides in the Center of Biotechnology of Sfax, Tunisia, and the coordinator of the project SAFWA. It's my great pleasure today to say a few words in the opening of this webinar, and I see that there are several people connected with us. Let me start by thanking the coordinator and the members of the European project NextGen Biopest for this initiative of joint dissemination action for our both project. I also thank the speakers and the participants, all the participants. I invite you to contribute actively to the discussion by posting your questions on the Zoom chat, and we will also have a brief session of Q&A at the end of the webinar. Thank you. Angeliki, the floor is yours. Thank you.
Thank you, Dr. Ruiz. Let me share my screen. Hopefully you can see my screen. So good morning again from me. I am Angeliki Milioti. I'm an agronomist as a background, and I work as a dissemination, exploitation, and communication manager at Smart Agro Hub. So our today's webinar is entitled Biopesticides, Driving New Generation and Good Practices to Farmers. And let's briefly see the objectives of today's webinar. Give me 1 minute. Okay. So first of all, the main objective of our today's webinar, as it is obvious from its title, is to raise awareness and educate our participants about biopesticides. In other words, we are going to see the types of biopesticides, what is their role in today's agriculture, and how they differ from the traditional ones. What are the main differences between chemical pesticides and biopesticides? Second, through this webinar, we want to promote the integrated pest management methods. And we are going to do this through, first of all, introducing these methods to you and show how in this method, in the IPM method, we combine biological, mechanical, and other methods. Also, we are going to discuss about a major problem of today's agriculture, the insecticide resistance. We are going to see how the diagnostic tools that will be developed through our projects can improve the pest control strategies. And finally, we are going to connect the theoretical part with the practical part, because we are very happy to present some successful case studies from the field of IPM and biopesticides. So these are the four main objectives. And of course, all these objectives are related to sustainability, which is an issue that is very important for all of us, especially in the field of agriculture. So we are going to connect biopesticides and integrated pest management to sustainability, because all these things that we are going to discuss today will help in protecting the human and environmental health. We are going to discuss how these methods preserve biodiversity, how the agricultural productivity will be enhanced through these practices. And also we are going to show how we will align our practices with the agricultural trends that show up in today's world. Let's have a quick eye on the agenda. Now we just completed the first part, the welcome and introduction. As I said, we will continue now with a pre-webinar quiz. It will be held through Mentimeter. I will show you exactly after the agenda how we are going to do this. Then we are going to have some speeches from distinct members of our projects. Dr. Mavridis, Dr. Vondas will explain the challenges and the technologies that are used in pest management. Then we are going to have some brief presentations about the projects in the context of which today's webinar is organized. And after that, Dr. Ferrer and Fonte Francesco are going to give some talks about the success stories and the farmers' opinions on IPM. After that, we are going to have again a post-webinar quiz in order for you to see if your knowledge has been enriched through this webinar. And then we will conclude this webinar with a Q&A session. So give me 1 minute to share the quiz with you. I think you can see my screen again. So please scan this QR in your mobile, or you can join at menti.com and use the code that is displayed here. So you can join the quiz. Let's wait 1 minute for all of you to join. Please confirm if, okay, I see some hands here. Nice. Yes, I see a raised hand. Okay, so let's start. First question, what are biopesticides? Please choose your correct option, and we will see how the answers are formed. Next question, what does IPM, Integrated Pest Management, primarily focus on? Which of the following is an example of a biopesticide? What is a key advantage of using biopesticides in pest management? Why is insecticide resistance a growing challenge in agriculture? How can molecular diagnostic tools help in pest management? What is a key characteristic of RNA-based pesticides? What are Bacillus thuringiensis-based pesticides? And now we completed the first quiz. Thank you very much. And now we will proceed with the first session. It is entitled Understanding the Problem, and I give the floor to Dr. Mavridis.
Many thanks, Angeliki. I will start by sharing my screen. Sorry. Okay, so now hopefully you can see my screen now. Is it okay?
Yes.
Okay, great. So, hello everyone, and thank you for joining. I'm Dr. Konstantinos Mavridis from IMBB 4th, and today I'll be talking about insecticide resistance, a very problematic phenomenon, and how it affects pest control and agriculture, and how modern diagnostic tools can help us manage this growing challenge. One of the biggest challenges in pest control today is insecticide resistance. At the same time, the number of available pesticides is decreasing due to stricter regulations and high development costs. This combination makes pest control more difficult and puts sustainable agriculture at risk. To put it in simple words, if we don't manage insecticide resistance effectively and immediately, we'll have fewer tools left to protect our crops. In this simple image shows how insecticide resistance develops and spreads. Firstly, when applying a pesticide, most pests die after treatment, but a few resistant ones survive. These survivors reproduce, passing on the resistant trait to their inheritance. With repeated spraying, the number of resistant pests grows until the insecticide stops working. This is why when we overuse the same chemicals over and over, this makes pest control harder over time. But why are insects so resistant to insecticides? It is true that insects have developed multiple mechanisms to develop resistance to insecticides. Some avoid insecticides by changing their behavior. This is called behavioral resistance. Others, they reduce absorption of the insecticide by modifying their outer layer. This phenomenon is known as altered penetration or cuticular resistance. Some insects survive exposure through target site resistance, which is caused by genetic mutations in the target site of the insecticide, which makes insecticide active. And finally, the fourth most studied mechanism is that the insects can use detoxification enzymes like P450 esterases to break down or neutralize insecticides before these reach the target site and take effect. This process is called the detoxification resistance. So indeed, in the field, we have seen some extreme cases of resistance over time. I put an example here which is a great example from Tetranicus urticae and acari, which affects many crops. And we have seen that some populations of this acari have developed multiple resistance to several different insecticides. Here we see a case from a population from Greece, which we detected in 2020, where resistance levels indicated by the resistance ratios were extremely high, and not only that, they were extremely high across multiple active ingredients that belong to different mode of actions, including abamectin, clofentazine, pyridabend, and many others. So, since fewer insecticides are available today due to high development costs and strict regulations, as we mentioned earlier, we can see that very easily in this graph, where it shows that the number of active substances from 240 in the 90s dropped to just 90 in 2020, while at the same time, the cost per development of a new insecticides has surged past 340 million. And of course, with policies like the European Green Deal, this trend we expect it to continue and get even worse. So, what's the solution here? The obvious one and the one that is very well discussed nowadays is to address this phenomenon via investing in biopesticides. This will be discussed in the next talk by Professor John Vondas as part of the Next Gen Biopest project. But at the same time, and additionally, we can apply insecticide resistant management, or IRM, to extend the lifespan of the chemicals we still have available. So, to keep current insecticides effective, we need the evidence-based insecticide resistant management. IRM is crucial because it helps us extend the lifespan of existing chemicals and ensures that they continue to provide effective pest control. A key thing in IRM is monitoring resistance in order to manage it. Monitoring resistance is essential and effective for IRM, and why this is important? Because detecting resistance early as possible helps us prevent its spread by applying targeted and evidence-based control strategies. And how do we do this? We have two main approaches here, each with its own advantages and disadvantages. These two approaches are the classical bioassays and some modern approaches like molecular diagnostics. Bioassays are used to test how insects respond to insecticides, but the disadvantage here is that they can be time-consuming, impractical, and have low sensitivity in detecting resistance. On the other hand, molecular diagnostics detect specific resistant mechanisms quickly and accurately with high specificity and sensitivity, but they require specialized equipment and expertise. These tools are complementary rather than one replacing the other. And with advancements in technology, the disadvantages of molecular diagnostics that we mentioned earlier can be overcome because molecular diagnostics are becoming faster, more practical, and more widely available, making resistance monitoring at the molecular level more efficient and more easy than ever. So here at IMVB4, we follow two key strategies to study and then tackle insecticide resistance. First, we identify and validate important biomarkers of resistance using functional validation tools, either in vitro or in vivo. Second, we develop and apply robust, accurate, and cost-effective molecular diagnostics to detect these biomarkers in the field. This allows us for a quick and reliable monitoring of resistance. So by combining these approaches, we aim to improve resistance detection and help design better pest control strategies. And I'll tell you in the next slides how we do that. A great example of why it is important to determine the value of resistant biomarkers using the functional validation tools either in vitro or in vivo. Here is an example where we studied two mutations in the ryanodin receptor gene, which is the target site for diamide insecticides that are widely used against several pest species. We see here that the G4946V mutation provides very high resistance, making diamide insecticides like flubentamide and chlorantraniliprole 100 to 200 times less effective. Meanwhile, another mutation in the same gene, termed Y4790M, has only a minor impact in the phenotype, causing a much lower level of resistance. And this shows why we can't just assume that all mutations have the same effect and all markers have the same effect. Validating these biomarkers helps us understand which mutations truly drive resistance, so we can target and develop diagnostic tools for these specific alterations through resistant management strategies. So once we have identified these important and key resistant biomarkers, the next step is to develop these effective molecular diagnostics and use them to detect resistance in the field. A key concept here is early detection, because as early as we detect detection, the better. This helps farmers and pest control professionals choose the right strategies before resistance spreads, and this can happen very quickly, as we have observed. Our goal here is to create practical and field deployable diagnostic tools that can be used efficiently and at a low cost, making resistance monitoring more accessible and effective. So this is very ideal. When we target to develop these diagnostics, we faced a problem, which is that a major challenge is that in developing these molecular diagnostics, we didn't know which techniques to target for. There's a variety of available techniques, each one with its own advantages and limitations. There are PCR-based techniques, sequencing-based techniques, or isothermal-based, which are not always easy to implement and integrate in a standard workflow. We aim here to detect different traits like mutations in the DNA, overexpression of genes. So we need to find methods that consolidate these detections and make the approach as practical and as scalable as possible. So we came up with two diagnostic solutions. The first one is called the lyophilized RT-qPCR assays, which is like assays in a pill-like format, which you can see here in these tube strips. And this approach has several key advantages. It does not require neither RNA or DNA extraction. You just simply add the sample, which is the lysate of the insect, the pest, or the pest that we target. It is very quick and field applicable. It can be used with portable qPCR machines directly in the field. And all the reagents are preloaded in this format, which minimizes hands-on steps and improves accuracy. The second diagnostic solution that we proposed is complementary to the previous one, which is field deployable. This is a lab-based solution, and it is based in droplet digital PCR. ddPCR is the third generation of PCR technology and is now considered by most as the gold standard for detecting genetic mutations with high precision and, most importantly, with very high sensitivity. It works by partitioning each sample into thousands of droplets where PCR reactions occur separately. This enables absolute quantification without the need of standard curves or calibration of the machine or whatever. And it detects very low frequency mutations with high accuracy. And when I say very low frequency and high sensitivity, it can detect resistance at extremely low frequencies, as little as 0.1%, meaning that we can identify one resistant pest, one resistant mutant insect in a pool of 1,000 wild-type ones. This, as you can understand, makes this method very practical because instead of testing insects one by one, it allows us to create very large pools of insects, up to 1,000 individuals, into one single sample. This dramatically reduces the number of tests needed, saving time and resources while maintaining accuracy and robustness. This makes ddPCR a powerful and scalable solution for large-scale resistance monitoring. To ensure the reliability of our molecular diagnostic methods, what we do first is to perform analytical validation, where we first validate these assays against gold standard methods that are regularly used in the lab, such as pool sequencing and individual genotyping. In this example, we see here strong correlations between our ddPCR diagnostics and the traditional gold standard sequencing methods, and this confirms their accuracy. And not only confirms their accuracy, it's a proof that it can replace or complement these methods, providing fast and more scalable resistant monitoring. Up to now, we have successfully applied this method in various species like Franklinellus dentalis, whiteflies, Bemisia tabaci, Tetranicus urticae, Panonicus citri, and many other pests, showing the method's broad applicability. But beyond analytical validation, we also confirm that our molecular diagnostic assays can accurately reflect real-world resistance, and this can be done through genotype to phenotype association studies. For example, we see here a very strong correlation in the Tetranicus urticae between a mutation, the H92R mutation, which causes MET1 resistance, and the LC50 values of MET insecticides like tebufenpirad and femperoximate. So by establishing these correlations, by establishing this relationship, we ensure that our molecular diagnostic assays are not just detecting mutations, but are also provide meaningful predictions of insecticide resistance in the field. But again, moving a step forward beyond simple correlations, I would like to show you a long-term study that is tracking intoxacarb resistance in two types of luta in Greece over the last 9 to 10 years. Here we monitor two target site mutations, F1845Y and F1848I. Both these mutations cause intoxacarb resistance, and we monitor these mutations using ddPCR alongside bioassays by monitoring the LC50 values of intoxacarb, which measure phenotypic resistance. So both genetic resistance, that is indicated by this blue line, and phenotypic resistance, which is indicated by the red line, have increased steadily from 2014, when we started this study, to 2023, where we stopped it. However, looking closely, genetic resistance, we can see that it appeared 1 to 2 years before phenotypic resistance was even detectable. Okay, so by 2019 and 2020, where resistance levels were high enough to cause field treatment failure, this phenomenon was not manageable, but mutations have already peaked 2 or 3 years earlier. So this study highlights how molecular diagnostics can predict indeed resistance in the field many years in advance, allowing for earlier intervention before control failures occur in the field. Our molecular diagnostics can also be used in different other scenarios, like to quickly monitor the resistance profile of invasive species. This is particularly important for Europe and Greece. Here's the example of the Spodoptera frugiperta, the former armyworm invasion, which happened in the summer of 2023. and poses a major agricultural threat. This pest is polyphagous, and when it invades, it quickly spreads and causes very big problems to crops. So we recognize this urgency. We quickly partnered with Bernanke Phytopathological Institute to assess the resistant profiles of these invading populations. So using our molecular diagnostics, we were able to complete a full resistance screening within weeks, monitoring key markers along 7 different insecticide classes. And this rapid response demonstrates how molecular tools can enable fast and effective resistance monitoring, helping to guide timely pest management decisions. Fortunately, we didn't detect resistance to most of the available active ingredients, so this can be used to combat the invasion of Spodoptera fujiperta. So to summarize, our work has advanced insecticide resistance monitoring by validating key resistant biomarkers through in vitro and in vivo tools, and developing robust molecular diagnostics based on these biomarkers. These tools have been successfully applied in real-world cases like long-term monitoring in the case of Tuta absoluta, and invasive species control like the Spodoptera fujiperta invasion. However, these tools are already available, but is not available at the time is policy guidelines, which are still needed to fully integrate these molecular diagnostics into insecticide resistant management programs in order to become widespread adopted. So moving forward, the challenge here is to bridge the gap between research and policy, making molecular diagnostic tools a standard part of pest management programs, which is still yet to be done. So before closing, I would like to thank all our collaborators and funding organizations who made this research work possible. It was carried out in collaboration with several leading institutes in universities across Europe, including Bernanke Phytopathological Institute, Kien University, the Agricultural University of Athens, University of Exeter, and others. And I'd like to point out that our work is supported by European Union funding through the SuperPest project that has already been completed and the ongoing NextGen Biopest project, but also from national initiatives. So thank you very much. A big thanks to our collaborators for the contributions, and also thank you for your attention. And I'm happy to take any questions if there are any. Thank you very much.
Thank you very much, Kostas. I think it was a very detailed and useful presentation. I would suggest that in case there are questions, the participants can add them to the Q&A box, and the speakers can answer them at the end of the webinar. So please feel free to add your questions to the Q&A box. And now, I think we can proceed with the second session. It is entitled Tools and Technologies for Sustainable Pest Management. Professor John Vontas will give the speech.
Okay.
We can see your screen now.
Okay, you can see my screen,
Yes,
Angeliki,
yes.
correct? Okay.
Yes, thank you.
Thank you very much for organizing that. Good morning, everybody. My name is John Vontas. I'm based in Crete, Greece, and I'm the coordinator of the project NextGen Biopest, Next Generation Biopesticides for the control of the most difficult to manage pests and pathogens in fruits and vegetables. This project started about a year ago. Actually, it's a kind of continuation of a previous EU project that we also coordinated here in Greece. That was the title was SuperPest, and this project includes many collaborators. It's a big consortium with a kind of long history, including several academic institutes such as Forth, INRA, IVIA, Elgo Dimitra, University of Kent, Fraunhofer, University of Catania, Exeter University, Western University Canada, and many companies as well, since this is a research and innovation project, area projects, such as Biopest, Magma, Greenlight Bioscience, and other research institutes. So you can see all the research institutes in these slides. I forgot, sorry, I didn't mention everyone, but this is a really very nice consortium, including also stakeholders and end users, such as cooperations and prefecture representatives. The objective of the project is to develop innovative products and methods and integrate this with existing approaches to achieve effective, environmentally friendly and sustainable protection against the most difficult to manage pests and pathogens in fruits and vegetables, and enable uptake by the end users. More specifically, we will try to develop novel and innovative methodologies, not having in mind to incorporate them into advanced integrated pest management schemes, so that to combine the more effective tools with what we already have in sustainable way. And the main problems that we have identified are several, are mainly arthropod pests, such as whitefly, aphids, and mites, some of which also transmit pathogens and virus in particular. We will focus on major fruits and vegetables crops. Pesticide resistance is a major issue, and we have also, based on the initial analysis, targeted those difficult to manage pests, arthropods, and also some major pathogenic diseases. We have in preliminary background work, we have measured that most of the chemicals are used against those pests and crops. So the specific objectives of our projects are listed in this slide, and I will briefly read them. Include, okay, first we need to capture a forum of end users and try to understand what are exactly the problems that they would like to include in the project. Then we have some technical specific objectives, including the development of molecular platforms, diagnostics for pests, pathogens, and their traits, for example, insecticide resistance mutations. To develop novel biological control agents and devise strategies to enhance their efficacy in the field. Of course, there are several products already in the market, but we wanted to have improved, for example, parasitoids or other beneficial insects capable to have a great performance also in the presence of chemicals sometimes, or green chemicals, or to perform also in the difficult resistant varieties of vegetables. Sometimes we know that this is a problem. There is some incompatibility between resistant varieties and biological control agents. To develop, validate, and bring to the market novel RNA-based pesticides. This will be achieved in collaboration with Greenlight Bioscience company, a big company based primarily in US, with headquarters also in Europe, in Spain in particular. And they have developed such products for against both mice and fungi in the States. So the idea is to further test those products and further improve these products and adapt to European grapevines and other crops. So specific objective 5, to develop and validate novel resistance inducers, highly selective juvenoid and repurposed green chemical, all together in either biostimulants or green chemicals that will replace chemicals in integrated pest management schemes, with hopefully equal or equivalent performance and efficiency, but very low residues in the crops. Specific objective 6 to optimize agronomic and ecological practices to increase the resilience of agricultural production against biotic stresses and enhance sustainable IPM in fruits and vegetables. Agronomic and ecological practices are becoming very important in Europe. For example, using certain approaches and ensuring the presence of parasitoids or predators in certain crops and fruit tree orchards. That's extremely important to keep an ecological balance and again reduce substantially use of chemicals, but enable pest control. Specific objective 7 to integrate the next generation biopest outcomes with existing methods into novel experimentally validated IPM concepts. So the idea is not only to suggest protocols for improved IPM, but measure the outcome both in terms of performance, efficiency, cost, socioeconomic acceptability, and other parameters, and also demonstrate this improved IPM SEMS in the field. This is extremely important also because the project, as I said, it has innovation features. We need to reach the end user to maximize its impact. Analysis of social, economic, and environmental aspects by University of Slovakia. And finally, to promote integration of knowledge dissemination, exploitation, and uptake to end user. And this communication activity that we have today is towards that direction. This slide shows briefly what I mentioned already, and it has been included in the specific objectives, the translation pathway of the innovative plant protection product development. As I mentioned, biological control agents, novel biopesticides, agroecology practices, new diagnostic and automated platforms to support evidence-based plant protection. All those developments in the red arrows will be developed, tested, and validated. The most efficient one will be included in advanced integrated pest management SIMS via synthesis and field trials, and so that at the end of the project, which will last for 4 years, we hope to have fully validated novel biopesticides, novel concepts and practices in IPM, advanced capability in Europe for testing, for example, or developing novel biopesticides also against additional pests than the ones that have been included in this project, and the intervention concept for many alternative crops. For example, if we demonstrate that the double-strand RNA pesticides are useful and can replace chemical insecticides in certain crops and pests, including the most difficult to manage pests, it will be, we hope, the next step might be easier to demonstrate the efficacy also against the additional targets and crops. And this is the PER chart showing the different work packages that have been included in the next generation biopest project: diagnostics, biological control agents, RNA-based pesticides, low-risk pesticides and green chemicals, agronomic ecological practices, synthesis into IPM, demonstration fields, extremely important component of the project. It's something that is missing in several countries, particularly Southern Europe. It's more advanced in Central and Northwest Europe, but not so much in countries like Greece, for example. Socioeconomic and environmental assessment, regulatory issues for the novel tools that we will develop. and the communication, dissemination, and exploitation. I can tell you briefly, if I have another couple of minutes, some of the initial outcomes. We are running into the end of, towards the end of the first year of the project. There is still another 3 years. So, but I would say that we have some actually not very promising results from Work Package 5. Many low-risk pesticides, green chemicals that have been developed in other regions such as Australia. Some of them are in the market, some of them are at early stages, are, for example, plant extracts, combinations, mixtures. They are not very effective against the difficult to control highly resistant pests that we deal with in this project, unfortunately, so this is a concern. However, at other ends, such as the biological control and the RNA-based pesticides, it seems that we have more potential. There is more successful stories. For example, the Green Light Bioscience products on double-strand RNA that have been tested now in America and Europe, also with contribution of our project, but also funded by other sources in US. Among them, the mildews project was undergone an extensive trial field testing program in more than 150 trials. Highly, very successful overall in terms of equivalent efficiency with so this GLP RNA formulations has equivalent efficiency with fungicides that have been used for the control of grape powdery mildew. And this is a very promising outcome. It will, of course, substantially facilitate the registration of those products also in Europe. They have been registered in US now, and it's great that this project, Next Generation Biopest, brings Green Light Bioscience very close to European farmers and hopefully the application of those modern double-stranded RNA pesticides, no residues in the environment, highly specific, no insecticide resistance. We hope that this can be expanded to other pests, including arthropod pests such as mites. There is a lot of ongoing research, of course. There are sometimes it's case specific, it's pest specific, the efficiency of those double-strand RNA formulations. For example, it seems that they are not so efficient against Lepidoptera species, but they are against two-spotted spider mite, as you can see in this slide, and the next one, where it has been shown that those products are efficient against both Tetranychus urticae and particularly Panonicus citri, which are very well controlled with those double-strand RNA products compared to chemical pesticides. And finally, I would like to mention that we also have success stories in the enhancing biological control agents. As I said, the aim is to both expand their spectrum as well as advance their performance and fitness in terms of varieties and presence of pesticides. So with among biological control agents that we work, and this is led by Biopest, by Tolis Pekas, and several research institutes, Institute Agro, INRAE, Elgo Dimitra, IVIA, and University of Catania contribute to those tasks, which include searching for new enemies in different places in Europe or Africa or other places, evaluating their efficiency and fitness, as I said, in different resistant varieties. And also under insecticide pressure, low insecticide pressure in IPM SEMS, include Phytocide mice, Triflodromus reki, with potential for controlling spider mites and other mites in tomatoes. Predatory thrips of the genus Franklinothrips to prey spider mites and thrips. T-date mites to control tomato rust mite while feeding on powdery mildew. mirids of the genus Phyllophorus to control mealbugs, psyllids, aphids, and wild flies. And finally, parasitoids of the genus Anargyrus to control the invasive Mediterranean mealybug Phenacoccus solenopsis. So hopefully in the next, in another meeting in the near future, we will have more data to present. Kostas Mavridis in the previous presentation already presented some of the developments in the diagnostic technologies that have been also included in this pest. So thank you very much for your attention. I would be happy to take any questions if you have.
Thank you very much, Professor. We will collect the questions in the Q&A box, so they will be answered at the end of the webinar. Thank you very much once again. I think that now we can proceed with the project's overview. Dr. Suad Ruiz, would you like to start with the SAFGA project?
Yeah, thank you. I will try to share my my presentation.
Yes.
Do you see my presentation?
Yes.
Okay, so I can start.
Thank you. Yes.
Thank you. So I have already introduced myself. I'm the head of laboratory of biopesticide in the Center of Biotechnology of Sfax, Tunisia. Our project was submitted in the frame of PRIMA thematic area water management 2022. It's research and innovation action, and the project title was Alternative Biopesticides for Safe Integrated Pests and Water Management around Mediterranean, with the acronym of SAFWA. We started in the 1st of June 2023 with a grant amount of about 1,400,000 euros. This project was undertaken in the frame of an initiative that we started since 2009. It's to drive a local biopesticide based on Bacillus thuringiensis from the lab to the market. So we started with the first project in the frame of ASH 2020 to develop the product, and then we continued with PRIMA to progress in the TRL of this product. Just to remind quickly that BT, Bacillus thuringiensis, is a safe and natural bacteria that we can find in soil, and it's active.
Sorry, yeah. Dr. Ruiz, are you changing your slides because we see the first one, or you have not started yet changing the slides?
I changed, but it seems that you are not seeing the... I changed, and now? Do you see now?
Now, yes.
Now you see? So
Can you change again the slide? Okay, now it changes. Okay.
Okay, so I come back to this one. As I said, BT is a safe and natural bacteria that is used to protect crops against pest larvae, and it acts through a crystal of delta-endotoxin that is, when it's eaten by the larvae, it provokes the paralysis of intestine. It's the insects stop feeding and finish by death. The aim of SAFWA is to release to the market an innovative solution combining a new competitive biopesticide to cultural training aiming to reduce land and water pollution through new agriculture practices. So we will drive to the market new cultural practices to farmers in 3 experimental farms through the Mediterranean, in Tunisia, Spain, and Turkey. These biopesticides are based on 2 strains, one from Tunisia and one from Lebanon, and they will be used in the field assays to treat olive, citrus, and pomegranate trees, as well as tomato, to protect these different cultures against 5 pest species, Tuta absoluta, Press C3, Press oleae, Phyllochnistis citrella, and Ectomyelis ceratonia. We are 10 partners in this project from 7 countries. So we have, and almost the most of these partners were also in the previous consortium. So we have from Tunisia, we have CBS, Center of Biotechnology of Sfax. We have CTE, which is a technical center for the field essays. We have also a company, Medis, who will produce and market the product later. From France, we have TBI from INSA Toulouse for the development of the product, the bioprocess. We have from Spain, we have Biotech Med in Valencia, and we have also IRTA. And I am happy and honored to have my colleague Marie-Thérèse Ferrer today as speaker. We have also USG, which is a lab from a faculty in Lebanon. We have also UniSG from Italy, and also my colleague my colleague, Dr. Fonter Francesco, also is a speaker today. And we have Biance from Italy, from Turkey, and finally GKI from Germany. The main objectives or the specific objectives of our project are developing a new competitive alternative based on Bt biopesticide, validating large-scale production and formulation of the new biopesticide, reusing waste bioprocessed water by recycling it in formulation process, reducing the use of harmful chemicals by enhancing the use of biopesticide, implementing good practices in farming activities, and raising awareness among farmers about good practices impact on water and land safety. So in the previous project, IPM for citrus, as you see, we worked on the value chain of the product, from the beginning, the upstream to the downstream. We worked also on the proof of concept, on the activity optimization, and on the economic maturation. In the current project, SAFWA, we focus mainly on the communication with farmers. So as you see in the WP2, we will continue or finalize the optimization of the bioprocess and the development. In WP3, it will be field assays, efficacy, and regulation. And in the WP4, we will focus on the farmer culture practices. And I guess that Michele will give some details in his talk later. For the WP2, as I said, industrial bioprocess and product formulation, we focus on optimizing the production and formulation process and prove the efficacy under controlled laboratory condition against the model insect Spodoptera ferrugiperda, and we will optimize the efficacy and its reliability under various standard environmental conditions of one optimized formulation. For the WP3, which is the heart of the project, if I can say, with WP4, we will work on the molecular identification of the strains to show their safety, a preliminary efficacy analysis, evaluation of insect toxicity at lab scale, evaluation of the side effects of the formulation. We already showed in the previous project that our product are safe against human, animals, and beneficial insects. And the 5, we evaluate the efficacy of the formulated products under commercial growing condition in crop grown in Mediterranean open fields. And finally, we will prepare information for a dossier to present the for regulatory purposes that could lead to the developed product authorization for field use. As you see here, and as I said, we have 5, we target 5 pests, and we will treat 4 plants to show the efficacy of our products. So the relevance of SAFWA relies on its double focus on farming good practices and innovative solution to reduce land and water contamination by chemical pesticide. The project originality mainly lies in a new approach based on an educating program for encouraging farmers to adopt new farming practices. And the WP4, as I said, is dealing with farmer cultural practices and civil society engagement. I will let later Michele will give more details about this WP4, I guess. And finally, for the WP5, communication, dissemination, and exploitation, our ambition is to disseminate our findings among different stakeholders, including researchers, public-private sector, company leaders, and R&D decision maker, public authorities, especially for policy guidelines, and also potential final users, mainly farmers, ONG, governments, private, individual, and so on. At the end, or our ultimate goal is to exploit this product, so to be able to market the products through our partners, which is Medis, the Tunisian company. And thank you for your attention. If there are any questions, we will let them to the end as we agreed. Thank you.
Thank you very much. Thank you for the insights of the SAFWA project. And now let's proceed to microbiopest. Juan, can you hear us? Can you share your screen?
Hi, everyone. Can you hear me?
Yes, yes.
So let me share my screen. Okay, can you see my screen?
Yes.
Okay, so today I'm going to give you an overview of the microbiopest.
We can see your screen, but now the screen is on the webinar. We don't see the
okay,
PPT.
okay. One moment. Let me share. Can you see it now?
Yes.
Okay, good.
Okay.
So, perfect. Today I'm going to talk about the overview of the microbiopest team, and which involves the discovery and development of microbial biopesticides that includes both bacteria and fungi to control vectors of diseases and agricultural pests. And it's important to mention that this is a project that started back in 2023 at the Institute of Molecular Biology and Biotechnology, IMBB, 4th in Heraklion, Crete. So the project outline is based in 3 main phases.
Sorry, we don't see the,
yes.
The slides. I don't know if you change your slide now.
I just changed my slide.
We are stuck on.
Can you see
now it's changed. Okay, thank you.
Hold on, let me see if I set again the presenter mode. If I change, can you see the change?
Now we are on the second slide. I don't know if you changed again.
Okay, so I think it's better if I keep it this way. Can you see the next slides?
Yes.
Okay, I think I'm going to present like this.
Okay.
Can you read the text?
Yeah, I think yes. Maybe you can increase the zoom.
Let's see, probably like this. Okay.
Yeah, yes, thank you.
Okay. So this project is divided into three different phases. First, we have the discovery phase, the research and development phase, and the commercialization phase. The first phase, which is the discovery, involved the field work. So we went to several places in Crete that I'm going to show you in the next slide. And we collected samples of different nature, for example, soil samples, rhizosphere, dead insects. We wanted to look at different sources where it was very possible that we could find microbes that could have insecticidal activities. Then after collected the collection of the samples, we isolated each of the different morphologies of bacteria and fungi that we obtained. And then we established a microbial library. And then we tested the insecticidal activity. So for the first part, we visited 65 locations across Crete with the different environments. And then the next step was to check the insecticide activity. So we did a preliminary screening because you can imagine that this included a high throughput of a big amount of bacteria. So we checked about 730 different bacterial isolates plus 49 bacteria. And then we assayed them for 48 well plates across 7 days. And we selected those isolates that were efficient at killing 100% of the larvae. In this case, we used Culex pipiens within 3 days. So that was an indication that these were the most potent biopesticides. After selecting these isolates, the next step was to scale up the bioassays. So instead of starting from a small volume of biopesticide in a 48 well plate, we scaled up the production on the bioassays to test the toxicity. And then we also use different steps of inactivation, because something important to keep in mind is that if we want to develop a product, then it's very important to use inactivated bacteria that can be heat inactivated, air dried, also called desiccated, or sonicated, so that we can extract the metabolites that are causing the mortality to the insects. And then the next step that includes the research and development includes the insecticidal, to understand the insecticidal mode of action, to check the activity spectrum. And it's very important to keep in mind that there are some of these secondary metabolites or protein toxins that are produced that might be very specific to a type of insects. A good example is the crytoxins or from Bacillus thuringiensis, or the binary toxins from Bacillus sphericus that are more specific towards lepidopterans or dipterans. So it's important to assess the different types of targets. And also it's important to check the off-target effects in beneficial insects, for example, pollinators, and see what would be the toxicological effects of these biopesticides. For example, to check what the sublethal doses, sublethal effects when they are released into the environment. It's also important to elucidate the nature of those molecules. So we conducted extractions using different solvents that would separate between polar and non-polar compounds that would give us an idea of the chemical properties of these compounds. And so here you can see, this is the map of Crete, where there's the 65 different locations where we did the sorting and collections of the samples. And it's important to mention that when we established the microbial library, we ended up with 195 samples that were collected. And from each of these samples, we collected a total of 1,709 bacterial and fungal isolates. And as you can see, this is a pretty exhaustive bacteria collection. So we only investigated 730, and from these we ended up with 17 with good or promising insecticidal activities that are now being included in the next steps for further elucidation of the toxicity. We found 46 fungi, all of them were tested, and from them, 9 showed a promising insecticidal activity, which were also included for the following steps. And now I would like to introduce you to the microbiopest team. We have the ERA chair, Professor George Dimopoulos, and the project coordinator is John Pontas. We have other principal researchers, and as you can see, this is a group that contains several postdoctorate associates, PhD students, undergraduate students, and there's different managers that are technical and financial. And with this, I would like to open you for any questions. Thank you.
Thank you very much for this insightful presentation. I'm asking again from the participants to add their questions to the Q&A box, and we will address them at the end of the webinar. And now, in the 5 minutes we have left for this session, I will share with you an initiative that took place in the context of NextGen Biopest project. I think you can see my screen. I will pass very briefly the overview because Professor Vonda has already gave a very good overview of the project. So I would like to focus here on an initiative that, as I said, took place in the project. A point that differentiates this project from others is that we have integrated an innovation hub on our website. And actually, an innovation hub is a virtual community, let's say, a virtual repository, because at the same time, it gives access to knowledge relative to the project, but also it helps us network further as stakeholders. So if you visit our website at this address, you will see the button Innovation Hub. And if you click on the Innovation Hub on the Explore category, you will see the material which you can access through your registration. So you log in. It is very simple to log in through a Google account, through your email. So please try it, and you will gain access to upcoming relevant workshops, job openings that may interest you, or also some relevant courses that are scheduled for the next months. So I encourage all of you to register to our hub. Until today, we have more than 40 people registered, and imagine that the initiative just started in the previous months. So this result is very promising. But also, in case you want to join our hub as a stakeholder and not as a person, we invite you to send us an email to angeliki@smartagrohub.gr and send us your logo of your company or your organization, and you will join our stakeholders forum. Also, in case you are working on a relevant project, again, feel free to contact us. You will find all contact information on our website. And until today, we have more than 30 stakeholders that have joined our hub, and I see some of them that are also participants today. So by joining our hub as stakeholders, you will have some benefits. Let's say you will be invited to our future events. It is very possible that you will be invited also as speakers in case we will organize a webinar relevant to your expertise. But of course, the main benefit is that your logo will be showcased among other relevant stakeholders, and you will increase your visibility in the sector. So this was a very brief reference to our hub. And now we are exactly on time to proceed with the next session, session 3, evidence-based IPM practices. And I give the floor to Dr. Maria Teresa Martinez Ferrer.
Good morning. Thank you for inviting me. My name is Maria Teresa Martinez Ferrer, and I will present you integrated pest management, evidence and practices. Okay, let's begin introducing to IPM. Integrated pest management is a dynamic, I would like to put this.
Sorry?
I'm sorry. Integrated pest management is a dynamic and flexible pest management approach that seeks to minimize the impact of pests effectively and sustainable using a combination of biological, cultural, chemical, and biotechnological methods. This is not working.
Maybe you can...
Yeah.
Now it moved, okay?
Now yes, thanks. Thank you. Integrated pest management, it's not working well. I don't know
Maybe
why.
you should exit the presenter mode.
Okay. Integrated pest management combines multiple strategies to control pests. So rather than relying on solely chemical pesticides, IPM emphasizes in prevention, sampling and identification, quantifying the pest population and identification of pests and natural enemies, determining when pest population require intervention through the threshold levels, studying the biological cycle of the pests to determine the optimal timing for treatments, whether chemical or biological, applying a combination of biological, cultural, biotechnical, and chemical strategies, and the evaluation and adaptation, continuously assessing these strategies. Okay. Let's dive deeper into each of them. For IPM, it's crucial to assess the presence, density, and distribution of pests and their natural enemies, and also whether pest populations have reached levels that require intervention. For that, we count on several methods, such as direct sampling, counting insects on leaves, stems, or fruits, using adhesive traps, pheromone traps, or light traps to capture insects and measure their populations, and technological devices, such as sensors or drones. Here we have an example. For sampling, the Lotococcus aviae in citrus is a new challenge in our citriculture, since this insect attacks small fruits, producing fruit drop, deformation, and decreased size in very small fruits. So sampling starts in petal folds and until that the fruit has 3-4 centimeters of the diameter. That is when the insect do the main damage. Samplings are made weekly from April until June, and we sample 6 to 8 fruits per tree in more than 50 trees. And if the threshold is reached, that is 10 or 12% fruits with mealybug, then we have to treat. There are different control methods that allow us to design control strategies in IPM. Let's go into them. Preventive measures. to avoid the spread of pests are important. For instance, do not move between farms wearing the same work clothes, and thoroughly clean work tools, and very, very careful with the collection boxes. There are several agricultural practices that prevent pest outbreaks. Crop rotation, growing different crops in a particular field season helps break pest life cycles. Cover crops that can attract beneficial insects that help to control pests. Planting resistant or tolerant varieties. Proper planting time to avoid peaks of pest activities. Field sanitation, such as removing crop residues. Soil health management. Optimizing plant spacing, water management, weed control, and trap crops. I would like to go deeper into the cover crops. They are very important in improving biological control, since biological control is one of the fundamental pillars of the integrated pest management. Maintaining a cover crop, this is an example in citrus, whether planted or spontaneous, increases plant diversity in citrus orchards. And this results in a greater diversity of predators and parasitoids that can improve the pest control. Biotechnological control combines natural processes and modern technologies, using technological tools like pheromones that are chemicals released by insects to communicate with each other, and usually are in IPM are used for mating disruption. Since by releasing large amounts of synthetic pheromones, males are confused and cannot locate females, reducing the pest population over time. The sterile insect technique, when large numbers of sterilized male insects are released into the environment, and these sterile males mate with females, but no offspring are produced. So over time, this reduces the population. The insect traps are designed to monitor or control pests together with pheromones or other tools. Here we have an example, again, for the Letococcus averiae in citrus. In this example, thanks to the identification and isolation of the sexual pheromone, devices have been designed that can be used for the detection, monitoring, and control the Letococcus averiae. These control devices consist in sexual pheromone and epinephrine. So the males are attracted to the pheromone and die upon contact with the insecticide. As the number of males decreases, there are fewer mating with females, leading to a decline in the population. And other biotechnological control is the advanced biotechnological techniques like CRISPR gene editing or RNA interference that are being explored for pest control. Biological control, one of the most important pillars in IPM, uses predators such as Coccinellidae, Chrysopidae, spiders, birds, parasitoids that are tiny hymenoptera that lay eggs inside or on the body of the pest insects, pathogens, including bacteria, viruses, fungi, and nematodes to control pests. Biological insecticides are formulations containing living microorganisms or their products. For instance, Bacillus thuringiensis is used as a biological insecticide against caterpillars, Diptera, and Coleoptera, and these insecticides are generally safer for humans, animals, and beneficial insects compared to the chemical pesticides. This is an example. One of the greatest success in biological control was the introducing.
Και ένα-δυο δωμάτια από εκείνο το κτίριο που έχει πάλι κάτι μικρά
Sorry?
γραμματικά.
Sorry?
Someone opened his microphone, but it was
Okay.
by mistake, yes.
Okay, sorry. One of the greatest successes in biological control was the introduction of Rhodolia cardinalis in California citrus against Etheria purchasii in the late 19th century. Severe infestations of Etheria purchasii were devastating the citrus orchards by feeding on plant sap and causing significant economic losses. Rhodolia cardinalis was imported from Australia, where it was a natural predator of the pest. The beetle quickly adapted to its new environment and effectively reduced Etheria purchasii population. This classical biological control set a precedent for future biological control programs worldwide. And currently, in situ, the conservation of Rhodolia is the best way to control Etheria purchasii. Chemical control plays a crucial role in IPM programs, especially when other methods are not sufficient. Therefore, we will make a controlled and strategic use of pesticides to minimize negative impacts. We have to use the pesticides selectively. The timing of application is crucial. So, because chemicals should be applied when pest populations are most vulnerable. Using formulations with low toxicity to non-target species. The efficiency of the application is essential for effective control, so the application equipment must be properly calibrated, considering the target pests, the product to be used, and the characteristics of the plantation. Very important, the resistant management, since overuse of pesticides can lead to develop resistant pest populations. IPM recommends rotating different classes of pesticide varying mode of actions. And now let's go to case study showcasing successful implementation in IPM practice. In my lab, we have been working for many years on developing integrated pest management strategies for citrus. This is a very dynamic agroecosystem, such as new pests are constantly being introduced and new active ingredients emerge, which require the definition of new strategies. In citrus, we have several aphids, several problems such as aphids, red spider mites, whiteflies, scales, medflies affecting citrus production. And sometimes the pesticide is high, and many times there is an insufficient control. The solution is to use all the tools that we have in IPM that we commented just as sampling, biological and biotechnological control, cover crops, precise application of plant protection products, timing, enhancing biodiversity, and with this we can reach more than 30% reduction in pesticide use, also effective pest control, improvement in citrus quality, and increased sustainability in production. Another example is the fight against Bactrothera ole, the olive fly that attacks and damages olives. And the solution is to use mass trapping, several traps in the field, lure with food attractants to capture females. And this diminish the population in the field, and it results in reduction in pesticide and better fruit quality. This is a control strategy of Theroplastes sinensis in citrus. Theroplastes sinensis produce tree weakening and sodium mold. And the solution is, first of all, studying the biological cycle. And we see that there is only one generation per year. Then we sample females that give us the population density. And if it exceeds the threshold, then we determine the optimal moment to treat. That is the final eclosion, the maximum first and second instars, and when there is no third instar. In this moment, we can apply just one treatment per year, and we will have, with this, we can combine this with release of natural enemies, and there is a reduction in pesticide use and better fruit quality. There is a problem applying pesticide because many times there are losses due drift and runoff when apply the pesticides. The solution is to optimize the dose and the spray volume adapted to the crop, the pest, and the product and the equipment we have, and also to use drift reducing nozzles. With this, we can have a 22% savings on pesticides and also a 27% reduction in soil coverage because of the drift. In tomato, they have aphids, red spike mites, whiteflies affecting tomato production with high pesticide and use and resistance. And the solution is to combine also the tools that we have in IPM, early monitoring and detection, biological control, cultural management, and precise application of plant products using drift reduction technologies. And this can result in a 40% reduction in pesticides, effective pest control, improvement in tomato quality, and increased sustainability in production. IPM give us environmental benefits since protects biodiversity and reduces pollution, economic benefits because it needs lower input costs and better marketability, and social benefits since it's safer for farmers and consumers and enhance food security. In conclusion, IPM is a proven sustainable pest control method. Combining multiple strategies ensures long-term success. Education and incentives can increase adoption. And a call to action is support IPM for a healthier planet and sustainable food systems. Thank you very much. And if you want to make some questions.
Thank you very much for your presentation and the case studies you shared with us. In case there are questions, we will address them at the end of the webinar from the Q&A box. So now we can proceed with session number 4, Farmer-Centric Insights from Dr. Fontefrancesco.
Hello, I'm just trying to share the screen somehow.
There is a button next to
No, the problem is PowerPoint is not the screen. But anyway, okay, at worst, you're going to see this one. Okay, anyway. Good morning, everybody. Sorry for my lame attitude with technologies. Okay. So here the question is posed on the actual use of biopesticide in the market by farmers. So this paper, this presentation is about the attitudes and bottleneck we find in the Mediterranean region, where all the projects are working, and also quite interesting area because of its composition that is split between EU countries and non-EU countries. So the research was conducted in 3 areas, in 3 countries. One, Spain, the Ebro Delta, in Tunisia, the Ebro region, and Turkey, the Adana province. What was the question here? The question was how farmers perceived and use, if they use biopesticide in the area, and what are actually the bottlenecks, as I said before. To do that, we conducted an extensive research in 3 areas. That it was quite quantitative research based on surveys, and particularly semi-structured interviews conducted in 2 rounds in Spain, 26-22 interviews, 50-50 in Tunisia and Turkey, 12-15. These numbers may appear small for researchers that are more into quantitative studies are quite the standard in terms of qualitative research, so don't be surprised. As I said, the data collection was focused on the pesticide usage, farmer awareness and perception by pesticide, and factor influential adoption. So what is the main result, the key findings? Well, actually, we see limited use of biopesticide, but this decision is mostly based on economic constraints, regulatory framework, and the market demand. These are common trends that we see also in the literature, but we see also quite a distinct attitude in each of the regions. So just to give you an idea, this is the areas, and these are the three main areas of research. And what's the situation? Well, Spain. Let's start from the EU countries. In Spain, thanks to the work of agronomists, the work of research institutions, for sure the legislation, EU legislation, there is a higher awareness of the existence of biopesticides. Many have used it, maybe as a biostimulant more than pesticide, but still they have idea of the existence of this product. However, they highlight still the high cost of this product, the limited availability, limited availability in quantity, but also in temporal availability. So it's not a consistent presence in the market. So if you have to plan an entire campaign just on a product that you are not sure how to preserve, and the other hand to have it, for sure it's a problem. The other part is the skepticism regarding the efficacy. So these are the key issues that we have to keep in mind, mostly concerning for sure commercial partners, but there are other elements that are pushing into the direction of biopesticide. In particular, is the EU legislation and the increased demand for products that are somehow sustainable or can be traded as sustainable. Both the things are somehow limiting the use of synthetic pesticide and opening a market for biopesticide. Tunisia. We see a moderate awareness of biopesticide, and most of the informants still rely on chemicals and synthesis of pesticide. The economic constraints in terms of the cost of this product are highlighted as a fundamental part. But on the other hand, when we see awareness, is awareness connected with export. So not linked with the internal market, but the possibility of exporting product in the EU. For sure, also in Tunisia, we see the government regulation is moving towards these new forms of these new tools. The other part that is particularly interesting for our informants is the integration between traditional knowledge and traditional practice and these new products that are closer to what they perceived, their way of farming. And for sure, the interest of their interest for the possibility of export. Finally, the Adana. Here we have seen the lowest awareness in the existence of biopesticide. Still, most of the farms rely on conventional pesticide and remedies. They highlighted lack information, risk of aversion because of novelty of this product compared with their practice. And still the reliance on agronomists and consultants that still suggest the use of chemical solutions. What is moving towards a alternatives is not regulation, but on the one hand, market. Let's not forget the importance of export, and the other hand, the higher resistance to chemicals of pests. So we see, coming to a conclusion, a scenario that is quite varied, but we should consider all these different factors. For sure, there is a question concerning regulation, and national and international regulation are important, but still, the differences here is about education, awareness. So, first of all, we have to realize that the adoption of biopesticides is largely hindered by the economic fragility of farms, specifically small and medium farms that are still the majority of farms across the Mediterranean region. The other hand, if we have to explain it in simple terms, the farmers cannot afford to risk to miss an harvest. So they want to avoid the risk. So this is crucial in terms of communication, and in particular, communication about how to use properly this product. There is a widespread skepticism, and the other part is the lack of availability on the market of this product. For sure, the regulation can play an important role in supporting the chain, and this means in both incentives and subsidies, in terms of supporting people to be less afraid of the risk of changing remedies or methods of farming. Education programs are important, and even more important is the creation of networks, local networks, where to share knowledge concerning how to use and what are these products. For sure, the collaboration between farmers, producer, researcher, and policymaker is still a crucial element that should be implemented more if we want to see a substantial change. So where we are going? Unfortunately, let's say biopesticides are still a potential alternative. It's not real in terms of the perspective of farmers. There is interest, yes, there is interest because of the possibility in terms of sustainability, but still there are many elements that limit the use. So what we need, well, we need to support farmers, first of all, with training and knowledge, but also with the financial incentives. Fragility is there. We cannot forget it. And indeed, with your research, should consider very carefully the question concerning accessibility, even more than efficacy somehow, because of if we want the transition, farmers should be really aware of the efficacy of the remedies, but above all, they can be sure that they, every day, whenever they need, they should find in the store the products. And this is not the situation we see at the moment. So I'm open to question, and I thank you all for the attention and the time for this presentation. Thank you.
Thank you very much for your presentation. It is very useful to see the farmers' opinion in this topic. And now we can proceed with the post-project quiz. I have included in the chat the URL you can visit and the code, but also you can scan the QR that I will show you now. So there are two ways of joining, either scanning this QR or visiting this page and add this code that I have included in the chat. So let's start. First question, how do biopesticides differ from synthetic chemical pesticides? Which of the following best describes the role of IPM? Some people have already answered. Which of the following is not considered a biopesticide? What is a major benefit of RNA-based pesticides? What is one key advantage of implementing IPM strategies? How do molecular diagnostic tools contribute to sustainable pest management? Why are case studies of successful IPM implementation important? How do Bacillus thuringiensis-based pesticides contribute to protect from chemical pollution? And this was our last quiz. You can self-assess your knowledge before and after the webinar. And now I don't know if Dr. Ruiz would like to give some key conclusions of today's webinar. I don't see any questions from the audience. You are muted.
There are no questions for speakers?
I guess no, I don't see anything in the chat or in the Q&A.
Okay, so it means that all what we presented was clear, so enough clear. Okay, I am happy to see that through the quiz, almost all the answers are right. So our audience is aware about the advantages and additive advance of the biopesticides. Okay, to summarize, we can say that we went during this webinar, we went through many alternatives of chemicals pesticide, like IPM or next generation biopesticide for diagnosis and treatment. And also we see a bit with Michele how farmers perceive new alternatives and around Mediterranean, and the chance for these farmers to adopt these new alternatives. Of course, if they, because they are aware about the advantages of biopesticide, but they need especially financial incentive. And as said Michele, the main gap is about regulation. So there are the main or the key points that we retained in this webinar. At the end, I would like to thank all the audience for their time and input, and thank you also for joining us today, and hope seeing you in next events. Angeliki?
I would like from my side to thank you all. Thanks to all speakers for their expertise and insights. All projects website, all 3 projects, in order to stay updated. And there you can also find the contact details for all of us. So since there are no questions, thank you all. Have a nice day. You will find the recording on our YouTube channel after the end of the webinar.
Thank you very
you.
much, Angeliki. Thank you very much, Suat, for organizing.
Thank you very much.
Bye-bye.
Thanks.
Thank you.
Bye. Thank you.
Bye-bye.
Congratulations.
Bye.